Welcome to our last session of day one. The title is Awards, Window or Just Window Dressing. Awards are meant to showcase great accomplishment and also to inspire others to follow in these footsteps. Here we highlight two perfect examples, and they will tell us if it's just window dressing. We will learn how each of them has been impacted by prestigious recognition and how they link this success with the team and career that have brought them to this place. We'll hear from Pamela Björkman from Caltech, who is the 2021 Green Guard Prize winner, and from Natalie Gomez-Ospina, who is at Stanford Bio-X and the winner of the 2021 Rosalind Franklin Medal from the Genome Writers Guild. Please be sure to click on their bios on the conference website. They are long and impressive. Also, be sure to read about the origins of the Green Guard Prize. Dr. Paul Greengard, Nobel Prize winner, named the prize after his mother, who died in childbirth, giving birth to him, and he donated his Nobel Prize winnings to create this award. Now let's hear first from Pamela and then from Natalia. Thank you.
Thank you very much, Carla. It's a real pleasure to be here. And I'd like to start off by saying that Rosalind Franklin has been my heroine ever since I started doing science in the biomedical world. And so when I came to graduate school, I decided that I was interested in structural biology. And so I was interested in things that would apply to, say, viruses or immune responses to viruses, or with the thought that maybe you could use structural biology to try and help somehow human health. So that's been my interest ever since then. Now, when I started graduate school, there weren't that many women in structural biology, especially in the United States. I think the UK had a series of women, including Rosalind Franklin and Dorothy Hodgkin and other women who were very prominent structural biologists. The United States at that time, which was the late 70s, early 80s, was behind in that sense. And so it was an extremely male-dominated field. And I ended up reading everything I could find about Rosalind Franklin, which included Ann Sayer's book, Rosalind Franklin and DNA, and then The Eighth Day of Creation by Horace Freeland Judson had a section on the discovery of the structure for DNA. It was a kind of fairly balanced section. Brenda Maddox's book, Dark Lady of DNA. I'm probably forgetting some here that I've read over the years. I also saw Photograph 51, which is about the people involved in the race for the structure of DNA. And I have to say that, like, from everything I read, Rosalind Franklin didn't think of it as a race. She was in it for the science. She was not in it for awards and recognition. She was extremely successful in everything she undertook, starting with fundamental research on structures of coal, moving on to work on DNA, which is what most people know her for now, about her now. But after that, she went and worked on fundamental structures of viruses. So I've just been so impressed with what she accomplished, and the fact that she actually died at 37, but made major impacts in 3 fields, continues to really inspire me to this day. And especially her ability to work under what must have been incredibly difficult circumstances. So I've always been really inspired by her. So that's my introduction. I just want to talk about the thing that excites me most, which is our research into trying to use structural biology to actually make a vaccine. And the type of a vaccine that I would like to make is a vaccine that might prevent another epidemic or pandemic. When the pandemic started, at least in the US, we became aware of it in about, it became very real to everyone in March of 2020. And at that point, we were working with Michelle Nussenswag's lab at Rockefeller University to solve structures of antibodies they had isolated from human convalescent COVID-19 donors. And we solved those structures of these neutralizing antibodies bound to the coronavirus spike trimer. And these are some examples of structures that Christopher Barnes, who was then a postdoctoral fellow in my laboratory, solved using cryo-electron microscopy, which, and so a lot of these would be near and dear, these techniques, to Rosalind Franklin's heart. Christopher is now an assistant professor at Stanford, by the way. Okay, so I don't want to go into structural detail about these, but that's the coronavirus spike trimer. And the spike is, of course, what allows the virus to get into a host cell. So that's the part that you see sticking out of the virus, and it gloms onto the receptor for itself, which is the human protein called ACE2. Okay, so you have to have that to get the virus in. And the spike is what binds to that. And so the spike is responsible for getting in. And so most of the neutralizing antibodies that you raise, if you're vaccinated or if you've had COVID, are against the spike. And in particular, most of those are against the so-called receptor binding domain, or RBD. And so Christopher did a number of structures, and these are all RBD binding antibodies because, for the most part, the strongest neutralizing response in both vaccination and illness, COVID-19, that is, if you've been infected with SARS-CoV-2, are against the receptor binding domain. Okay, so he went on using antibodies from COVID-19 convalescent donors from an initial study by Robiani et al. In the Nussenswag lab, and a later study by Vinci Wong et al. In the Nussenswag lab, where they isolated antibodies from vaccinated people. There's no real differences in the types of neutralizing antibodies that you raise in response. So that the vaccines are working like a prior infection, except of course you don't get sick and you have prior immunity, so that if you do get exposed, you can bring that up and then hopefully not get infected at all, or not have a serious consequence from the infection. So from those structural studies, Christopher and our lab defined 4 major places on the receptor binding domain that these types of neutralizing antibodies bound to. So we called them class 1 through class 4. This is a simplification, I have to say, because we're seeing things that overlap between class 1 and class 4, and so are other people. And so this was a compilation of our work and a number of other structural biologists around the world. And so we classified antibodies that we could get structures for. Okay, so I want to emphasize here that the class 1 and class 2 antibodies are very strong neutralizing antibodies, and they block ACE2 binding. So they block the receptor site on the receptor binding domain. So that means you can't infect at all. These are great antibodies, but the problem is that that's where you get the mutations in the variants of concern. And so we're all worried about Omicron. Most of its mutations are in the class 1 and class 2 area. So I'll get back to that. So in about March of 2020, we started reading the coronavirus literature, and it was very clear from what previous coronavirus virologists had written about is that they knew all along we were going to have another epidemic or pandemic. There have been 3 in the past 20 years. The first one was SARS-CoV-1, and the second one was MERS coronavirus, and now we're in the middle of the SARS-CoV-2 pandemic right now. These all have zoonotic origins, and the worry is that as humans keep encroaching on animal habitats, there's more and more virus transfer into humans. Most of the time, nobody even knows that they've been infected with an animal coronavirus, but every once in a while, something happens where we get an epidemic or a pandemic, and that's what's happened in this case. And we'd like to be able to stop that, because this was predictable that this was eventually going to happen after SARS and then MERS. So what Alex Cohen, who was then a graduate student in the lab, did was to use what we call a mosaic nanoparticle approach to potentially make what we think of as a possibility for a pan-sarbecovirus vaccine. So again, that means SARS-like beta coronavirus. We're not trying to target all coronaviruses because we think that probably isn't possible. I showed on the last slide, there are coronaviruses that cause common colds and so on, and they're sequentially different enough that it would be very hard to make a pan coronavirus vaccine. But what we want to do is prevent the next spillover from a SARS-like beta coronavirus. And these are commonly infecting people who live near bat caves, for example, in Yunnan, China. It's just that normally, so far, we've been lucky except for the SARS pandemic and the SARS-CoV-2 pandemic. Okay, so what he did was Alex started by going through the literature and he selected receptor binding domains from SARS-like coronaviruses of pandemic concern. He made a nanoparticle to which we covalently attached mixtures of different RBDs. So he made, for example, a mosaic 8, which means that it had 8 different RBDs from SARS-CoV-2 and 7 animal coronaviruses. And he tested that in mice, and he saw a cross-reactive antibody response that resulted in neutralization. So that was preclinical proof of concept. Okay, so I want to say why we want to make this mosaic particle. So the idea is that when you stimulate your antibody response, there are reactions in your lymph node, in germinal centers, for example, where you have B cells. That's what makes antibody. And they have B cell receptors on their membrane, and they bind to whatever. If it's a vaccine, they bind to that. If it's a virus, they bind to that. Okay, so let's say you were making a nanoparticle and you made it where you attached all the same RBD to the nanoparticle. So there are 60 sites on the one we use. So this would have, we call it a homotypic nanoparticle. It would have, say, 60 SARS-CoV-2 RBDs attached to it, and they're all SARS-CoV-2. So now when that goes in and binds to different B cells, they all have different receptors. The ones that bind most tightly will trigger the antibody response. And what you're going to get are really strong antibodies to what we'll call distracting epitopes. That's the orange triangle there. So you'll make great antibodies to things that are different from one strain of sarbecovirus to another. Okay, so that's not what we want. But what if instead you injected this mosaic nanoparticle here? The distracting epitopes between the different RBDs are now different. So all the colors of the triangle are different. And so you don't bind as tightly because antibodies have 2 identical arms and they can't reach between the distracting epitopes because they're all different on average when they're right next to each other. So what we're hoping in that case is that you would preferentially activate antibodies that bind to this purple part of the receptor binding domain. And that's the conserved part. And those are the antibodies we want because they would not vary in variants of concern. At least there's no evidence so far that they ever could. And they're in common between the various sarbecoviruses. Okay, so that's the theory behind this. So structurally, is there any reason to think this might be possible? So what I'm showing you now is over on the left. This is the idea here, is that you'd preferentially make antibodies that bind to the purple part, which is the more conserved part, by cross-linking between adjacent receptor binding domains. And then next to that, we're showing a structure of the spike trimer, and the receptor binding domain is there. It's been color-coded in a gradient of sequence conservation from variable in orange to purple is more conserved. And so you can see that the stuff on top is more orange. And that, if you look over to the right now, those correspond more to the class 1 and class 2 epitopes, which I said are really strong neutralizing antibodies, but they don't always work as you change things in the variants of concern, and they certainly don't work between the different sarbecoviruses. So there is a purple patch on the RBD, which is encompassed mainly in the class 4 epitope, and also to a certain extent in what we called the class 3 epitope. And structurally, Claudia Getty and Christopher defined a structure of the type of antibody that we really want to raise. This is something that's called CO22. It's isolated from a COVID-19 convalescent donor from one of Michelle Nussenswag's studies. And these are not commonly raised during infection, but they're there. So the idea is our vaccine would trigger this type of antibody more. You can see it's mostly class 4, but it reaches up towards the ACE2 binding site, which is at the top of the RBD there. So it actually blocks ACE2 binding as well as binding to the more conserved part. So those are the antibodies we want to get. And so the question is, can we get those? So what I'm showing you here in this movie is you're going to see the spike rotating. The receptor binding domains are in yellow, and you're seeing like a cartoon diagram with alpha helices, beta sheets, and so on. And so this is the RBD can either be up or down. It has to be up to bind ACE2. And what you're seeing there are the conserved and not conserved parts, orange, not conserved, purple, conserved on the RBD. So you can see what we're trying to target there. And we were showing it to you in the context of the trimer. And we have some structural examples. We and other laboratories and companies have examples of these types of antibodies. So we want our vaccine to raise exactly those types of antibodies, and we know it's possible. Okay, so what Alex did, to go through this really fast, is he went through the literature from a paper from Vincent Munster's lab at the Rocky Mountain National Labs, and they had categorized sarbecoviruses of potential pandemic concern, and he made these nanoparticles as either homotypic, so all SARS-CoV-2. He made a mosaic 4A, which is the first 4 listed there. He made a mosaic 4B, and then in mosaic 8, he combined them to make mosaic 8. And then critically, we didn't include all of the RBDs that we've actually made because we separated out 4 to look for neutralization and binding to so-called mismatched strains. So this is an important control. Are we getting cross-reactive responses? And in particular, we left out conventional SARS, that is SARS-CoV-1, because that would represent a virus that could spill over at any time. We don't know what's going to spill over, right? So we have to use things that we already know about. Okay, so we left 4 out to look for mismatched responses. And so those are the 4 that we left out that we could use as controls. So this is published now, so I'll refer you to our paper from about a year ago. But what I'd like to say is that in unpublished work, we have now isolated monoclonal antibodies from the immunized mice, where we saw more cross-reactive responses to mice that were injected with the mosaic 8 versus the homotypic. So now with the help of Miso Park and John Williams at a nearby research institution, City of Hope, they've isolated monoclonal antibodies, and we've characterized them with binding and neutralization assays. And the really interesting thing is these antibodies here, as examples of monoclonals that were raised in mice, they appear to be what we're looking for. That is, they bind. On the left, you're seeing under EC50, those are binding responses. And then on the right, for each antibody, we see the neutralization responses. So the lower the value, the better. So we're seeing very powerful antibodies that actually neutralize at low concentrations. That's what we wanted. But perhaps more importantly for us, we're seeing broad cross-reactivity across these different strains of sarbecoviruses, including some that are mismatched. So I wanted to show you that what we're trying to do now is improve that vaccine composition with this antigen design cycle. So basically what we do is we vaccinate some kind of experimental animal now, and we look in the serum for the polyclonal antibody responses using competition assays, using a technique called polyclonal electron microscopy, and then doing serum neutralization assays. And then we isolate single B cells from which we get monoclonal antibody sequences. We solve 3-dimensional structures of those antibodies bound to the spike trimer. And then we do neutralization assays with the monoclonal antibodies. And from that, we have this design cycle to try and improve perhaps the mosaic thing. But let me just show you that so far, with this limited set of monoclonals that we've isolated to date, it looks like we are getting the types of responses that we want. And I'm going to show you some structural data now that was done by Cheng Fan, who's a relatively new postdoc in the lab, who's solved at least 30 or more of these structures so far. And then she solved all 7 of the monoclonals that I showed you. And we're going to just show you where they bind. So here is Cheng Cheng, and on the left there, that's where ACE2 binds in the orange part of the RBD. So that's the less conserved part. And now I'm only showing you 6 of the structures. She solved the 7th one, and obviously we need more monoclonal antibodies and more structures and so on to draw more firm conclusions, but it's looking really good so far. The epitopes are in the cyan dotted outline here. And these are mapping mostly to these purple regions. So we're really excited about that. We think that the approach we're doing is working as planned. And I just want to point out that one of these antibodies here, which I'm sorry, I can't see it, but I think because the people are on top of this. Yes. Okay. That's the antibody that was not very cross-reactive. You can see that a lot of those strains are not filled in, meaning that it didn't bind to those. And that's one of the rare ones that we found so far that is not binding to the purple area. But we can definitely just biochemically and structurally distinguish this. So far, we need a lot more of this work, but we're very excited about the prospects of this. Okay, so, and this is what's really exciting to us is we're doing challenge studies in mice that carry a transgene for the human ACE2 receptor. These are done at Rocky Mountain National Labs by Vincent Munster and Neeltje van Dormelen. And so these are their results. They've injected these mice with either homotypic nanoparticles or the mosaic 8. And so what you see here is the result of so they were immunized, primed and boosted, and then they were challenged with SARS-CoV-2. So this is a matched strain, right? Because we have a SARS-CoV-2 RBD on both the homotypic, it's entirely SARS-CoV-2 RBD, and we also have one of the 8 RBDs on the mosaic 8 is SARS-CoV-2 RBD. And so what you're looking at on the left is, do the animals lose weight? That's a sign of that they're getting sick. And the answer is no for both of the mosaic, for the mosaic 8 that we made, and also for the homotypic. So the red and the blue lines there, and you can see on the right is the survival curve. Those mice all live. The mice that are injected with the nanoparticle alone, so that's the control, these die and they lose a lot of weight. Okay, so the other form of the mosaic, which I won't go into, doesn't work quite as well. So it's clear that there are ways to make better mosaics. So this one, the mosaic 8 in its original form, works better than the second form we did. But the really exciting thing to us is for SARS-1, that is a totally mismatched strain in the mosaic and is mismatched for the homotypic as well. You can see that the animals that are completely protected are the ones that were injected with the mosaic nanoparticle. That's the blue and the green lines there under the weight loss. The red one, the homotypic SARS-CoV-2 particle, the animals, 5 out of 6 of them died and they lost a lot of weight. And you can see that on the survival curve. So we are exhibiting protection with the mosaic nanoparticle, but not with injections of the homotypic nanoparticle. So in conclusion, we've demonstrated that the co-display of the SARS-CoV-2 RBD with other RBDs works. This is very exciting to us. So we can get neutralizing antibodies. So it works just as well. So the fact that you dilute out the number of RBDs doesn't affect your protection against SARS-CoV-2. But even more importantly for us, the mosaic co-display demonstrates advantages against it potentially emerging coronaviruses because we see neutralization of matched and mismatched strains, and we also saw protection against the mismatched strains. Okay, so these are more technical points, but together we think that a mosaic nanoparticle is the best. It's a really good way now to protect against SARS-CoV-2. It could protect against potentially emerging sarbecoviruses. And since the variants of concern, their mutations are in the same place that differs, the same part of the RBD that differs between different sarbecovirus strains, this type of a vaccine would work against the current variant of concern that we're all worried about, which is Omicron, and potentially it should work against any variant that comes out because they're all mapping to that same orange place on the RBD. So with that, I'd like to thank the people in my lab, Christopher, who's now a professor at Stanford, Cheng Fan, who's doing the structural biology now, Alex, who started this whole project and is doing the vaccine design and the experiments and so on. I didn't get a chance to talk to you about what Magnus is doing for a potential vaccine or talk much about my collaborators, but they're all listed on this slide. So I wanted to thank you for your attention. And that was hopefully somewhat of a comprehensible discussion of what we're trying to do research-wise.
So now we'll hear from Natalie Gomez-Ospina, and she will tell us both a little bit about her terrific research and her complicated journey to the success she is at now. Thank you so much, Natalia.
All right. Well, thank you for the invitation to speak, and excited to tell you about my research and my path, and to talk about the importance of awards for young investigators. They're especially important for young investigators, and so I'll stress that. So I'll start with my life to my research. The only thread, and when I talk about my life, is sort of time and I guess grit, very little genius, more like hard work, a little bit of luck here and there, but I guess always a desire to do my best, to do everything to the best of my abilities, and maybe that's why I'm here. So I was actually born in Colombia in South America. I was there until I was a young adult. I was born to a single mother. I didn't have a father in my life most of my time, and I didn't mention that before when I talked about my life, but I became a mother this year. I have 2 children, and I'm a professional. I have a pretty good job. I have a supportive partner and the financial resources to take care of them. So now I can see what she accomplished, being alone without financial support, without family support, having me. So probably the consequence of that was, a lot of independence, resilience. That came from that, and I found solace in school. I guess, like a lot of successful people, you find that in school you find validation, and you like learning, and so I'm still in school. I pursue an MD PhD, and I'm an assistant professor, so I am still learning. So we moved to the United States for personal reasons, and I spent the first 2 years actually learning English, and I worked in the restaurant industry. I was a busser and a waitress for a little while until I was able to start at the University of Colorado Boulder, which was my sort of local school, and got the financial aid to do so. I wasn't one of those people who had the privilege of like growing up thinking, I want to do this. I think, children and students kind of set their dreams and goals based on the opportunities that they have, which is something that I keep in mind when I'm participating in admission processes. So it was, I think that at some point I started college in Colombia because as an engineer, it was sort of the practical thing to do. But in somewhere after reading some books, I decided that being a genetic engineer would be really cool. But in the 1990s, when I started all this, it wasn't that didn't really exist. So when I chose a major at CU Boulder, I chose molecular, cellular, and developmental biology, and double major with biochemistry. That seemed close enough. And that I joined a structure lab and stayed realized that, if I want to do this, I need to be in the lab. And I worked in the lab for a couple of years and was very lucky to be in this lab. We had one of the few high voltage electron microscopes in the country, and we had a kind of a unique tool and able to participate in collaborations and get advice from people about, hey, you should probably go to graduate school. And then at that point I decided that I wanted to do in that spectrum from basic discovery to affecting human health. I wanted to be kind of more towards human health. So I decided to become a physician scientist and did my training at Stanford in the medical scientist training program. And even then I had some, this wasn't entirely my path to where I am today, was also had some twists and turns. I changed residency training, and I'm now a medical geneticist, which I think is an appreciated specialty in medicine, although more and more appreciated now that genetics is becoming more a bigger part in our lives, and people are starting to realize that it has diagnostic utility, and now even we can even start to think about treating people with genetic diseases, but it's one that a lot of medical students don't have a lot of exposure to. But again, it's another specialty where I get to learn all the time. I'm still a student. There are 30,000 genetic diseases. I will never know all of them, so I approach every patient as they have something to teach me, and I'm here to learn. And so in this work as a medical geneticist, I, with the skills that I had, I felt that the greatest value would be in developing therapies. And so that's what I do. And so I will, I think I can move on to talk about my research for a little bit of time, and then we'll talk about the award. There are multiple approaches to treating genetic diseases. My desire is to make treatments that are durable. It's hard to say curative always, sometimes, but at least as definitive as possible. And the approach that I've taken is to try to maximize the ability of the hematopoietic system to treat non-hematopoietic organs. And so I'm trying to merge regenerative medicine with now the new tools of cellular engineering in order to make very precise genetic medicines. And the beauty of using the hematopoietic system is we have 3 decades of experience transplanting it. And that, just a cellular infusion will fully reconstitute a functional bone marrow, as opposed to, stem cells for other types of organs. And in my practice, the diseases that, sometimes when I say my medical geneticist, I get a sad face from people. And I think that they do that because they feel like I'm always telling patients bad news, and that I'm usually don't have a solution from them, which isn't always true. But these are there's a group of diseases that really, really affect me. And these are neurodegenerative diseases in children. So these are children that are born healthy, that at either in infancy or within a few later can start to regress and lose neurologic function, usually relentlessly. And when you look at the genetic causes in particular, at least of what we know now, a lot of these diseases are caused by deficiencies in lysosomal genes. And this is also important for adult onset neurodegeneration. And so, for example, these particular gene GBA, which encodes glucocerebrosidase, and carriers of this it's sort of the greatest genetic risk factor for idiopathic Parkinson's disease. So what we not only do we think is important, I think that it's important to try to manage these children better, but also will have huge implications for our understanding of neurodegeneration in adults. And so these lysosomes are these organelles within cells that are responsible for catabolizing macromolecules in cells. So cells are better than people in that they recycle things back to other parts of the cells. And within these lysosomes, there are many types of enzymes, but also the membrane proteins that are necessary for these to function properly. So there is a large group of conditions, and about 2/3 of them will have some sort of neurological involvement. The beauty of the system, even though some of the diseases that these mutations result in are terrible, is that at least for the enzymes live within the lysosome, is they have this nice characteristic in the biology, which is that they are modified with mannose-6 phosphate residues, and they're constantly secreted into the extracellular space. And they can be basically taken up by any cell that has a mannose-6-phosphate receptor. And this, what means is that a cell that expresses enzyme can effectively cross-correct a cell in the vicinity that has the right receptor. So it's the property that is known as cross-correction, and is the basis for any therapy that's been designed for these types of disorders that involve lysosomal enzymes in particular. Although, you know, there's secretion, but they might be other interesting mechanisms that allow for this property of cellular cross correction. So there are some of these diseases have approved treatments. Not a lot of them. One is enzyme replacement therapy, which entails the regular weekly or biweekly infusion of a recombinantly produced enzyme. Sometimes in one approved therapy is intraventricular. So you can imagine that this sort of approach is quite involved in making an enzyme and then delivering this via infusions for the rest of their life with a commitment to spending 4 or 5 hours attached to an infusion device. And this means, you know, wearing a device in the head for the rest of their life. So another option is allogeneic hematopoietic stem cell transplantation, at least for some of the diseases, have been shown to be helpful. And the way that, so why hematopoietic stem cell transplantation for diseases that are not hematopoietic? And what happens is that one part of the therapy is that the bone marrow becomes a reservoir enzyme. So bone marrow cells, all of them will secrete enzyme, which can then be uptaken by some of the affected cells. So there's the enzyme reservoir part of it, but then there's also the function of these monocytes who can migrate to different kinds of tissues, and they become local enzyme factories. And so they can do what a recombinant enzyme cannot do. Particularly in the brain, we know that there's a process of extravasation and invasion that allows these cells to become microglia-like and reside for a really long time. And so so how do you leverage this type of therapy to these diseases, make it better, make it more effective, and make it viable for broader classes? So the first part of doing this is to make these transplants allogeneic. So we know that to go from an allogeneic transplant to an autologous transplant, allogeneic means using, you know, donor cells, and autologous cells means using the patient's own cells, but this means developing efficient strategies to make these, to correct the genetic defect or the biochemical defect. And we've done some work on several diseases to do this, and we have a platform to do it. The second part is, one fear that folks have, especially those who have clinical experience with this, is the pre-transplant conditioning that is required prior to transplantation. And so this is basically the administration of chemotherapy in order to create space and the niche for the new cells to engraft. So you can't just infuse the cells and hope that they will be there. You actually need to deplete the internal stem cell pools for these cells to go there. So it has pretty acute and also long-term toxicity. And so, especially in the brain and poor brain activity. So remember, many of these protocols are designed for bone marrow transplantation. So you measure the chimerism in the bone marrow as opposed to how much penetration did you get in the brain from this bone marrow derived cells. So we're working on autologous transplantation, and we're working on newer conditioning regimens that will make it more efficient for other organs, especially the brain, but also safer. So this is generally the platform that I designed for making autologous transplantation using genome editing. So as opposed to using genome editing in order to try to go after specific genes or mutations, we decided to do a safe harbor approach. And so the idea is to isolate the patient's own hematopoietic stem cells, engineering ex vivo. Right now, the platform, the tool of choice is CRISPR-Cas9, and we do it at a safe harbor, and I'll tell you why. And then the idea is that with repopulation, new hematopoietic system that can make enzyme, but also nifty monocytes and macrophages that can deliver enzyme to hard to treat organs. So why a safe harbor? So it allows you to, by removing the control of the endogenous promoter, you can control expression for to improve therapeutic efficacy. So we know that maybe achieving super physiological levels or recruiting certain type of cells could be more effective. And I'll give you some examples of how this works. We also know this is a better solution to the issue of allelic heterogeneity. So for many diseases, there are hundreds of mutations. So designing a genome editing approach, which is, takes a lot of work for every mutation, is really not feasible. And even sometimes for every gene, it's complicated. So with this way, we can address a whole class of diseases with the same with the same locus. You can engineer the coding sequences, which is also work that we're doing to give them better properties, enzymes with better stability, enzymes with ability to better cross the blood-brain barrier, etc. And again, this obviously is adaptable to many therapeutic proteins. And we chose CCR5 because of the high frequency of people that are knockouts, have deletions of this gene. It's about 12% in the UK Biobank. And the added benefit of that having not expressing CCR5 gives resistance to HIV. So we think of this as a well-proven safe harbor, or at least gene that can be co-opted for expressing lysosomal enzymes. So we've developed proof of concept studies for 2 lysosomal storage diseases, mucopolysaccharidosis type 1, Gaucher disease, and we have ongoing work on other lysosomal deficiencies. So I'll tell you with some detail of MPS 1 and a little bit of detail on Gaucher. So we started with this lysosomal storage disease. Again, this is a model disease because we want to show that the treatment affects many different organs. So you can see on the symptom column that these patients have cognitive impairment, skeletal manifestations, visceral manifestations. Pretty much every organ system is affected. And there is a severe and attenuated form. The attenuated form, the individuals that have this have less neurodegeneration. And this is important if you look down here at the bottom, is sort of the median enzymatic activity of these patients. So to have severe disease, you have to have almost zero enzymatic activity, but to have attenuated disease, even just 1 or 2% enzymatic activity prevents a lot of neurodegeneration. And this is, while these patients are affected, they're more mild. And so this tells us something about the therapeutic threshold that we have to achieve in order to really make a difference in the natural history of the disease. And even though this is a very complicated name and it sounds ultra rare, it really is an important disease because in many states, and I haven't updated this in the last 6 months, we're already looking for these children at birth in order to transplant them as early as possible to try to improve our outcomes. So we've already decided that in all of these states that we're to look for these children to treat them effectively. So these are some examples of how the editing works. So here we're using a constitutive promoter, the enzyme and the reporter that we use for our preclinical studies to track and isolate these cells are expressed all the time. So you can see that the hematopoietic stem and progenitor cells are all expressing the fluorescent marker. And we achieve about 35% correction efficiency. So 35% of the cell that we isolate for a patient would be corrected. If we add a marker like this, about 45-50% will be corrected when we use a more clinically relevant donor, which is just the enzyme without the marker. And because of this important role of monocytes, like I mentioned before, in macrophages, we can show that these human hematopoietic stem cells can make macrophages, and that these monocytes and macrophages express about 50-fold more enzyme than a normal cell. So these cells are expressing supraphysiological levels of the enzyme. For everyone working with genome editing for therapeutic purposes, we're always concerned about the safety. So we've analyzed safety in hundreds of transplants looking for tumorigenicity. We haven't found anything. And then we do also analysis of off-target activity for that site. So you can see, long story short here, we're doing deep sequencing of predicted off-target sites. The on-target is 90-something%, and a few off-targets come up less than 0.5% cutting efficiency, but they go away when you switch to a Cas9 that has higher fidelity. So very good off-target profile. And the beauty of starting with human cells, we didn't engineer mouse cells, we engineered human cells because we want to end up with a true or feasible candidate cell product. But to show their efficacy, we re-engineer the mouse model. So this is a mouse model of lysosomal storage disease that is made in an immunocompromised background to enable the human cells to engraft. And so we made this model. They asked the humans, there's no expression of enzyme, there's accumulation of tissue glycosaminoglycans. They have a skeletal phenotype. They have sort of subtle dysmorphology phenotype, and spleen and liver enlargement as well. So now we're in a position to say, does your treatment work? And you can see that transplantation, here's the biochemical correction. The urinary gags, which are a representation of visceral storage of the metabolite without the enzyme, and the kidney storage, decreases with transplantation of the edited cells. We have a decrease in the tissue gags. And in this particular graph, the brain was not significantly decreased, but I will go back to this, and there's partial enzyme reconstitution. But again, we're not trying to replace 100% of the enzyme. This platform we've adapted to other enzymes because that is the whole benefit of using the safe harbor approach. We're also using it to express the enzyme defective in Gaucher disease, which gives you sort of a spectrum of disease. The majority of patients are these type 1 that have visceral involvement, liver, spleen, and bone marrow. But then there's more severe forms that have infantile onset neurodegeneration. And then there's also the carriers of GBA mutations who have a increased risk for Parkinson's disease. So this is a very high interest of developing effective therapies for this disease. So this disease presented an interesting challenge in that the enzyme could not be expressed in the hematopoietic stem cell compartment. So as opposed to what I showed you with this other enzyme where we can overexpress it in all the cell types using a constitutive promoter, doing the same here would have prevented engraftment. So the safe harbor approach still offers a really nice solution, which is we just switch the promoter. So we engineer a small monocyte macrophage specific promoter, which is exactly where this enzyme is needed. And you can see that the undifferentiated cells with the constitutive promoter express the transgene and the reporter. With this specific promoter, there's very little expression, but with differentiation, then you can actually see expression of the enzyme and the cassette. And just to long story short, these enzymes engraft beautifully and make very nicely functional human monocytes and macrophages expressing the transgene. We've also there are some, while hematopoietic stem cell transplantation, I think it's great for a lot of diseases, it has some shortcomings. And it is particularly difficult for diseases like this one, Krabbe disease, where the neurodegeneration is happening so quickly that the effect of transplantation is too slow to make we use it, but we really want to get an effect in the CNS much sooner than what a standard hematopoietic stem cell transplant does. So this is a deficiency in galactocerebrosidase. In the infantile form, these kids are 3 to 4 months of age, and they rarely survive to the second year. Currently, the standard of care, if they're caught early on, which usually never really happens, and early enough to give them an allogeneic transplant, which only slows down the progression of disease. And the disease is also part of several newborn screening programs around the country. So what we're trying to do for this disease is say, okay, we'll do the standard autologous transplantation of engineered cells, but would also deliver some cells into the brain to temporize the brain, what's happening in the brain. So we have different sources of cells. So we've learned to engineer human neuronal stem cells, and we're also exploring the possibility of using cells derived from hematopoietic lineages that can be directly go into the brain in order to replace CNS macrophages. So I just wanted to talk, so the other part of this, which is the transplant conditioning I mentioned earlier, and our goal to try to complement on autologous cells with safer and faster brain-specific conditioning approaches. And so the current limitations are the toxicity of myeloablative conditioning, both in the systemic and the CNS, and how inefficient it is at CNS repopulation and slow, as I mentioned, for Krabbe disease. So we're working on targeting microglia specifically, and you can see that with a standard conditioning, and even with this, we see effects in the CNS. You can get maybe 10% of cells, bone marrow derived cells in the brain. With our new drugs that we're modeling, we can replace, these are the percent of microglia. This is the percent of microglia, or CD45 positive and CD38 positive cells that are now coming from the bone marrow. So we can effectively now replace most of the microglia and not only lower the dose of the bad conditioning agent, increase the efficacy in the brain. So we're moving in an excellent direction in terms of trying to maximize the ability of this engineer hematopoietic stem cell transplant to affect other organs, specifically the brain. So, that's very briefly, we use the hematopoietic system to deliver therapeutic proteins. As I mentioned, I'm very interested in lysosomal deficiency, but there are other therapeutic proteins they can be used for. The safe harbor is very flexible for the expression of these therapeutic proteins, and macroglia depletion can enhance efficacy and safety of hematopoietic stem cell transplants for neurological diseases. And with that, I like to acknowledge the top of these are sort of my mentors and sponsors, et cetera. And bottom here are some of the people, the very brave people that joined the lab of a young investigator. What I want to do now is talk about the award and the meaning of getting this kinds of awards. So, awards are important for multiple reasons. One is recognition, right? And it's very important for young scientists like me because it tells you you're doing a good job, you're on the right track, what you're doing. Sometimes you're not sure, are people really interested? You can be passionate, but you want that to be acknowledged. You want the award is also important for awareness of what you're doing, and for a practical part of that is part of what is needed to make it in academia and to be promoted is to make sure that people in your field, more senior people, are aware of you and are aware of what you're doing. So this is one mechanism to achieve that. An award also provides a platform to disseminate your research and your name, but also disseminate other things that are relevant to who you are as a scientist, as a female scientist, as a female minority scientist. So also a platform to be able to talk about that. And also, it allows you to be a role model. And I think that's super important for the Rosalind Franklin Society, that you're out there to be a role model for other people. So yes, although I would say one interesting twist of the pandemic is that some of those things, it's harder to know, the benefit of being the awardee and giving a talk and, again, as a young investigator and meeting other people in your field and having them remember you. I think it's probably a little bit less because of the platform that we're now doing, but maybe at the same time, it's a wider platform and then, things are getting recorded, more people are listening, et cetera. So we'll see. So very thankful for the award.
Well, thank you both. I think it's pretty clear that you are both incredibly deserving of these important awards, and it's very inspiring hearing both about your critical and current research and about your journey in getting there. And also thank all of you worldwide for logging in to day one of the Rosalind Franklin Society annual meeting. Please remember to join us again tomorrow for an exciting and inspiring agenda again. It will take you from photograph 51 to leadership in outer space. It's a pretty diverse set of speakers. You will find them all, again, exciting and inspiring. In the meantime, thank you all for listening, and certainly thanks to Pamela and Natalia for sharing their research and their experience. Thanks.